Hi everyone, I am Saniya Kule, and this is cute little Yangshi. And in this video, I want to talk to you about something that I really love and I want to share with you guys. And especially those of you who love art as much as I do, or those of you who may not necessarily know why art is so great. I want to talk to you about something known as bioconductor. What is bioconductor? And why is it so great? And why is it giving it an edge as well? So what is bioconductor, Yangshi? We're going to talk about what bioconductor is. So it is available on our statistical software. So I'm going to show it to you, and there are lots and lots of videos on these bioconductor packages as well. So basically, this is bioconductor. Bioconductor is basically an open source software for bioinformatics, and its mission of this project, so it's all free, and its mission of Bioconductor is to develop, support, and disseminate free open source software that facilitates rigorous and reproducible analysis of data from current and emerging biological assays. So they are a welcoming community of developers and data scientists, and they're all using the R statistical programming language, and it is open source, so it's available to all of you guys. It's open development, and it has 2 releases each year and an active user community. So they also have it as Docker images. So currently, as of this release 3.14, there are 2,083 different software packages. That is a lot, Yangshi. 2,083 free software packages. So there was a time when I didn't really know about Bioconductor. I was using packages, but I didn't even know about it. And then at the start of the pandemic, I kind of discovered this resource called Bioconductor. And I was really surprised and so excited because there's so many tools out there to help you with using computational tools and methods to analyze biological data for different research projects and biological experiment analysis. There's so much out there, and it's all available on R, which is so exciting. So what you're going to see here is that there's so many software packages. There's support on installing Bioconductor, installing R. I also have a video on installing R. But when you have finished installing R, all you have to do is if you don't have Bioconductor, all you have to do is you can write this code here, and it will install Bioconductor if you don't have it. Because basically Bioconductor, when you install packages in R, what you typically do is you just do like install.packages, and then you write the package name. But in Bioconductor, it's sort of different, because what you're trying to do in Bioconductor is you have to sort of call for these packages. You sort of have to install them using something called BioCManager ::install. That's how you install a package name on Bioconductor. And then similarly to how you load in packages in R, you can load in packages in Bioconductor. You get it, the R is like Bioconductor, like R. You just call library package name. So this helps you to install Bioconductor on R. And this is how you install a package on Bioconductor. And if it's not really going through, you might need to force the installation. You can just do force equals true, and that will like force it to install on your computer. Sometimes you may need to re-update some packages, or you just have to force it to go through for whatever reason. So this just forces it. And then when you have the package installed, you can just do library package name, or there's one of them called ABA Enrichment, which is a Bioconductor package. And then you can just you can load it in from the packages folder, or you can just do library and the package name. That's how you can do this. So I kind of showed you how you can sort of install packages right now on Bioconductor. But what are these 2,083 different software packages? Well, there's so many of these here. I would get so excited. You can just kind of scroll through these. It sort of was like a little gift for me to just sometimes spend time scrolling through these and seeing what all tools they have, Yankee. It was such a joy for me to see what they have out there. So basically what we can see here is that there are just so many tools. So many different tools out here. BioC version, iRanges, genomic info, genomic ranges. So representation of genomic intervals. So we can click any of these. And if we were to want to download this package, all we would need to do is the following. All we would need to do is, let's say we want to get genomic ranges downloaded. But if we want to get this package, what we have to do is just for genomic ranges, we just have to call BioC manager here. So we would just have to do BioC manager install, and then you have to put it in quotes, genomic ranges. That's how you install genomic ranges, basically. And then if you want to load it in, you do library genomic ranges. And one of my goals would be to have some package pushed up on Bioconductor. That would be pretty awesome. These are really high-end packages that make it there. So that's what you have to do. So because we're using this BioC manager to install packages in R, that's why we need to install BioC manager, because that's how we install packages like genomic ranges. We use this BioC manager and then install for package name. For instance, the package name here is genomic ranges. That's why we needed to install it. So what this is saying is it's quietly going to install it. If you need to have this package and you don't have it, if it's not already on your computer, on your R, installed in R, this is sort of how you would install Bioconductor. And then you can install all these packages that you need, for instance. And in fact, if we were to see genomic ranges, what it would tell us is that the way that you install it is also, you first see if you don't have BioC manager, if you don't have Bioconductor, you have to install Bioconductor, and then you have to install the package, and then you can load it in. And then in your R, if you want to browse vignettes, if you want to browse some information on these packages, then you can also just do browse vignettes genomic ranges, for instance. And what that would do is it's just going to give you more information on this R package. So I already have this kind of installed, but it's going to take you, if you do browse vignettes, it'll bring you here. An introduction to genomic ranges package, how-tos, a quick introduction, 10 things you didn't know, extending it, like all this information here. So they even have, sometimes some packages have this nice little vignette, which I really like. And they can tell you when they've updated it, and then they kind of go through and they give you some examples that you can run here, where they also say that if you don't have it, you have to install it on Bioconductor. You first install Bioconductor if you don't have it, then you install the package if you don't have it already, and then you load in the package. And then you can run through these tutorials here. For chromosome and ranges along chromosomes. So basically this package is focusing on efficiently representing, genomic annotations and alignment when you're working with high throughput sequencing data like next generation. So that's what this package is focused on. There are other packages that I was finding that I'm a huge fan of, like DESeq, which is helping you normalize data, find, gene expression data, find genes that are differently expressed between them. For instance, you can look at the HTML, this vignette. I really like these vignettes here. This was just updated a couple of weeks ago. What you'll do here is you can see how you can get help for DESeq 2, for instance. They have a lot of different examples, like how to get help for it, how to start with it, how to load in data as well, and genes and samples. What are p-values adjusted? These are all a bunch of tools out there. Log two-fold change, normalize counts. There are basically a lot of different functions and tools available out there to run your analysis. So people have written code and classes and functions and objects to do different things, to come up with heat maps, for instance. Heat map of the count matrix. You have conditions, paired end, single read, untreated, treated. How do you use their, and this is another library, pHeatmap, how do you use their tools to get different sorts of analysis results? Another one, I really love this one, is Cluster Profiler. They have HTML, sometimes they have a PDF reference manual available here, for instance. And then what you can see in this reference manual is they have a bunch of different functions with arguments and values that are returned, comparing clusters, information. You can look at these and run code using these examples and using David for enrichment analysis as well. So there are different tools here. Cluster Profiler helps you find out the functions of coding and non-coding genomics data as well. And it has a reference manual. Again, this always reminds you how to install a Bioconductor if you don't have it, how to install the packages like Cluster Profiler for Bioconductor. They have vignettes as well that we can just put this into the console. They have the reference manual. They have some news available sometimes. They usually have a PDF. Sometimes they have a vignette, which is really helpful because I really love the vignette. So this one is so helpful because they have a book down. And in the book down, they talk about, it's really well organized how you can use Cluster Profiler, how you can use it to find more information on genes, disease enrichment analysis. If you have a set of genes, this is another disease ontology database. You can figure out, you can take a set of genes, do enrichment analysis here. There's wiki pathways here, Reactome, there's KEGG. There's a lot of tools out here. You know, you can even view some of these KEGG pathways this way as well. You can visualize functional enrichments. You know, if you have a set of genes, you can visualize them. And these are all tools. What's really great is these are all tools that have been made to help you. And this is from the Cluster Profiler package as well. There's so many tools out there. So all you have to do is just go through and like download the ones that you need. Like there's Cluster Profiler, a universal enrichment tool for interpreting omics data. There's DOSE as well, disease ontology, semantic, and enrichment analysis. They have a reference manual as well that you can see. You know, you can see what this was just updated like 2 days ago. Similarity between some vectors, enrichment, DGN. What are the arguments a vector of onto gene IDs? And this is why I really stress gene IDs being so important. The onto IDs, p-value cutoff, different p-value adjustment values, you know, for multiple testing correction, background genes, are you mapping gene ID, gene name, like they have all these arguments. And I sometimes do prefer the, you know, vignette, which is in HTML sometimes because they give you better examples of how to use them. Sometimes they give you a full, yeah, they give you some full information here. There's KEGG, enrichment, wiki pathways, and you can copy their examples. I highly recommend working through their examples. There's a GWAS Tools package here. Sometimes they don't have a vignette, they just have PDFs, but we can look at the PDFs and whatever information they have provided. Classes for storing very large genome-wide association study, you know, like SNPs and p-values and annotation functions for cleaning and analysis as well. They have these things that you can look at, these GWAS tools here. Fisher's test, you know, fed up. This is a very cute name. I'm fed up of this. No, Fisher's test for enrichment fed, 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 for enrichment and depletion of user-defined pathways. Using Fisher's exact test, ran pathway analysis on custom annotations. So there are a lot of different tools out here. We can just keep on scrolling through and seeing. Summary statistics for rows and columns of sparse matrices, classes for single cell data. They have our package for visualizing tree and annotation data. We can check GG tree out as well. We can see the HTML. They have a tree data book. You know, exporting data, tree data, working with tree information, like phylogenetic trees. They have a lot of sources, KEGG graph, PCA methods, consensus. We can even look up any of these things here. They even have like ways that you can search assay domain, biological questions, technology, annotation, experiment, workflow. I can even look up enrich. If I want to do gene enrichment, in any way I can look up, okay, this is an enrichment package, fella, interpretation and enrichment for metabolomics data. I can see what this is over here. And quick start, I can look at any of these things. Load in the KEGG data, data objects, visualizing the results here. A lot of this stuff, page ranking. They have a lot of things here. Single nucleotide variant, same thing as SNP. Tools for conducting enrichment analysis of medical signature headings as well. Co-expressed genes is COGENA here. A workflow. They also have examples here you can run as a PDF. Cluster sizes. So yeah, there are just a lot of resources here. So many things, graph theoretic, RNA-seq data, identify genes associated with multiple expression phenotypes and single cell CRISPR screening data. There's so much out here. Yeah, there's just a lot. I always sometimes just spend time looking through these things. You can sometimes do Control F as well. You can also just see what all there is. There's so many tools. And there are also tools on GitHub too that that haven't yet made it to Bioconductor, but they are also equally good. And I can talk about those in another video. But yeah, there are just so many packages out here and they're all growing and they have to be maintained as well. There's so many tools. Feast, feature selection for single cell clustering. So if we had a problem, chances are that it could have been solved from Bioconductor already. Which is why I recommend seeing Bioconductor, seeing the tools, getting inspired by them, and just taking advantage of what there is. As Yangshi says, Bioconductor is the best. So there's so many things out there. You don't need to reinvent the wheel, but you can use some of these tools to help you with your work already. You can see what there is. If you're interested in like SNP, for instance, you can see what stuff there is. Testing of SNPs and SNP interactions. There's a trio package here. There are, a beta race SNP. There's just so much out here. Snipedia, query data from Snipedia, like it's like a Wikipedia, and provide some tools for downloading and parsing data, extract the most relevant information from SNPs. So they have this nice little Snipedia, for instance. You can download chromosome position, orientation, all of the stuff here, reference SNP ID, RSID, parsing functions, categories, is a SNP, is a topic, is a medical condition. There's just so much out there for you guys. So I hope that this helps. I'm going to be talking more about Bioconductor in some more videos, but I didn't know about this and I stumbled upon it. And I feel like a lot of people don't realize, and that's why R is still really cool because it has Bioconductor and so many ways to work with this as well. So I really hope you guys will consider Bioconductor for your research. And I will be talking about quite a few of my favorite Bioconductor packages as well. So please do look into Bioconductor. It is so powerful. These are all tools people are designing. There are also tools on GitHub available. So I hope that this helps you. I'm Saniya Kuller. This is cute little Yangshi. And I hope that you use Bioconductor. And please let me know if you have any questions, comments, any suggestions on key packages or anything in general. I'm always happy to help. Thank you guys and have a great day. And again, this is like an amazing gift. It's so exciting. I'll like to keep on top of the Bioconductor packages. It's so cool. And they do so many great things and tools. And they're going to help you make a difference in public health.